Please welcome Joshua Waterfall, Institut Curie, single-cell omics to characterize the tumor immune microenvironment.
Hello, everybody. Greetings from Paris. To start right off, or not, it is here. So first off, why do single-cell sequencing? It's not just technology for the sake of doing new technology and spending money. The first point is that clinical samples, especially biopsies, are generally very small. So if you can profile individual cells, this opens up whole worlds of clinical contexts that you can now address. The second point is that tumors are dynamic ecosystems of not only malignant cells, but all sorts of important microenvironment cells. And understanding these through bulk approaches is severely limited. Results over the last 5 to 10 years have shown that the microenvironment is a powerful therapeutic target. And these processes are not just genetic. This is a lot of differentiation and plasticity. So we have to understand, we have to take a multi-omics approach to understanding this process. So I want to make the point that I represent a small part of a really important team effort. So everything from the surgeons and the radiologists acquiring the initial samples to pathologists, immunologists, our sequencing platforms, that we all work tightly together. For those of you that recognize the left bank of Paris, this is the Curie, but we work very closely with Hôpital Necker, Gustave Roussy, and Montsouris to profile all sorts of patients with different types of cancer. We can do this in cancers that are very common, like non-small cell lung cancer, or very rare, like pediatric solid tumors. We can do it on we do it on adults, as well as children that are only 18 months old. And this is informative in cases where immunotherapy is entirely routine, and we want to know why is it only 30% of patients that respond, but also cases where immunotherapy is very investigative, and we want to know what would be the best approach to take in these patients. To show you a little bit of real data from a paper that's going to come out any day now in Cancer Cell, we've been profiling a rare pediatric tumor called rhabdoid cancer. And here I show you a merged atlas of 33,000 T cells that we sorted out of these rhabdoid tumors from young patients. So from this merged atlas, we can recognize what the cell types are, what the different T cell states are in these tumors, populations that are recognizing the tumor. We can break it out by the individual samples. And then most excitingly, we can identify the specific T cell clones that are recognizing that tumor. And this is where these projects move really directly into drug discovery. Because as we'll hear from other talks today, these TCRs can be powerful therapeutic agents when you reintroduce them into the patients. So with that, I will acknowledge my collaborators in rhabdoid tumor, and thank you all very much for your time.